Hi everyone, welcome back to my channel. I hope all of you are doing so great in your life. So in the previous video, we discussed about the prokaryotic cells, bacteria, archaea. So today we are going to discuss about eukaryotic cells and its cells or cell organelles. Okay, so let's start today's class. So without any further delay, let's start. All of you can take out your notebook so that you can note down the important points. What are the eukaryotic cells? Can you tell me? So eukaryotic cells are the new cells, you can say. Prokaryotics were primitive one, eukaryotics are the new cells, right? So that means they have a lot of things, a lot of cell organelles. They are so much independent of itself, right? So here you can see the eukaryotics were having a different domain, the third domain. And under the third domain, there was categories like the fungus, protista, plantae, and animalia. Okay, so all of these types of cells are eukaryotic cells. Eukaryotic cells could be organisms could be multicellular as well as unicellular. Multicellular, for example, us human beings. Unicellular like euglenoid. Okay, these are the eukaryotic unicellular or yeast. Okay, unicellular organisms. So let's discuss about the cell organelles. So in the eukaryotic cells, as you know that it is having cell organelles which are having double membrane. So double membrane means it provides extra protection to every cell organelles, right? And the size of the eukaryotic cells are also pretty much larger than the prokaryotic cell. So inside the eukaryotic cell, we can see a lot of different types of cell organelles. For example, the vacuoles, endoplasmic reticulums, ribosomes, okay, and all of these cell organelles are having their own functions, okay. First of all, why these are called as organelles? Do you know? So organelles means the meaning of organelle is little organs, okay, little organs. So basically, all of these structures, they perform their separate functions and independently they are performing their functions, correct? It's very similar to the organs, like we have stomach or liver or um, any large intestine or lungs, heart. All these organs are performing their functions independently and individually, right? So like no stomach is not going to help lungs, right? And neither like liver is not going to help stomach. So all of them are performing their own functions individually and independently. Similarly, these small little structures which are present inside our organ cell are also performing their functions independently. That's the reason they are similar like organs. So scientist has given name them organelles, means little organs. Okay. So what are the different cell organelles present? First of all, the cytoplasm is the jelly-like fluid. It's not the organelle, but it's the medium where all the organelles are present. Then we will be discussing about the main organelles, the nucleus, mitochondria, plastids, ribosomes, endoplasmic reticulum, Golgi apparatus, lysosome, vacuole, then the centrosome, centriole, then we will be discussing about cilia and flagella also, and at the last I will be discussing about the cell membrane. Why I will be discussing about the cell membrane at the last? Because I need to discuss about the active transport and the passive transport also. So it will be a like bigger topic. So I will be covering it in the last. Okay. So all of you get ready to watch a little video for a better explanation.
So, what are organelles? Organelle means little organ. Organelles are the specialized parts of a cell that have unique jobs to perform. Let's start with the nucleus, the control center of the cell. The nucleus contains DNA, or genetic material. DNA dictates what the cell is going to do and how it's going to do it. Chromatin is the tangled, spread out form of DNA found inside the nuclear membrane. When a cell is ready to divide, DNA condenses into structures known as chromosomes. The nucleus also contains a nucleolus, which is a structure where ribosomes are made.
All right, so let's first discuss about the cytoplasm. Cytoplasm is a jelly-like fluid. Okay, so it's a jelly-like fluid which is present all over the like inside the cell. So consider the cell like a ball, a hollow ball. Okay, so if you fill the hollow ball with water, what will happen? If inside the hollow ball there will be water everywhere, right? So it's similar to that structure. Okay, so cytoplasm is only the fluid content which is present inside the cell, and that also in between the cell membrane and the nuclear membrane. Okay, so these structure, these orange colored things are called as cytoplasm. Now, cytoplasm is very important. Why? Because first of all, it is providing a a home to all the cell organelles. So basically all the cell organelles are present in the cytoplasm. Okay, they are floating in the cytoplasm. All right, they are having a particular place. They are not floating from this portion to that portion, not like that. They are having that particular position only, location, but they are in a floating manner. All right, so that is the first function of the cytoplasm. Next function is, this is also the region for certain biochemical reactions. It's very important thing. For example, the glycolysis process occurs in the cytoplasm only. So it's important process, right? So that's why the biochemical reactions are also happening in the cytoplasm. Clear? So this is the first thing, which first part of the cell organelle. Now you can see that cell membrane, the genetic material, the cytoplasm, and also the ribosomes, these are called as the basic parts of the cell. Why? Because they are present in all the different types of cells. Okay, like they are, that's why they are basic part of the cell, or you can say functional region of the cell. Moving on to the next cell part of the cell, that is the cell wall. Now, the cell wall is present in the plant cell, in the fungus, and in the bacterial cell also. Okay, so animal cells do not have cell wall. So you can see that here you can see this is the cell and the outermost covering is the cell wall. So in the plant cell, the outermost covering is called as the cell wall. So this is providing a extra protection. Double covering means double protection, right? So animal cell do not have that protection. Okay, extra protection. So plant cell, in the plant cell, this cell wall is made up of cellulose, hemicellulose, pectin. Okay, or glucose. So various compositions are there in the plant cell. Here you can see the structure of the cell wall. This is the like hemicellulose. Here pectin is present, cellulose microfibrils are there, and lamella is there. So whole this complete structure is called as the cell wall structure. Clear? Now, in the bacteria, it is made up of peptidoglycan. Okay, so we have discussed this in detail in the previous lecture. Please go and see the previous lecture, that is lecture 1. Okay, next, in the fungi, the cell wall is composed of chitin, glucans, and proteins. Okay, so these are the different composition of the cell wall. But the main function of the cell wall is what? It's provide protection. It provides support, extra support, protection to the plant, bacteria, and the fungal cells. So it's a very rigid structure actually. It's the outermost rigid structure. Okay, and archaea also has cell wall. Correct? Archaea also has cell wall, but that's not made up of the peptidoglycan layers. Okay, it's absent there. Peptidoglycan is absent. So it's the function of the cell wall is first of all it provides support, mechanical strength to the cells. Okay. Then it also helps in the cell growth process. All right. So it basically helps in the formation of the cell plate in the plant cell. Okay. Next is the it controls also controls the diffusions, helps in the communication through the plasmodesmata channels. So plasmodesmata are like some channels, small little channels which are present in the like cell wall. Through these channels, any substances can pass from one cell to another cell. Okay, so that's the plasmodesmata. Next, it is of course it is helping in the defense mechanism. Like of course it's the outermost covering, so it is protecting the plant cell from any kind of pathogens. Okay, it also prevents the excess loss of water, and it also helps in some storage of some extra carbohydrates or energy. Okay, so these are the functions of the cell wall. Now moving on to the next cell organelle, the important one, that is the nucleus. And this is the principle of the cell because it's the head, it's the center. Okay, so nucleus was discovered by Robert Brown in the year 1831, and inside the nucleus, chromatin material is present. And Fleming was the scientist who stained the chromatin material and named it as a chromatin, basically the genetic material. Okay, so here you can see the structure of the nucleus. Here, outside the nucleus, there is a nuclear envelope is present. Okay, nuclear envelope consists of some pores which are called as the nuclear pores. Through these pores, the RNA and the proteins moves in and out of the cell. Not cell, sorry, nucleus. Okay, at the center of the nucleus, there is a structure called as nucleolus, and this nucleolus is helping to synthesize the ribosomes or rRNA. Okay, nucleoplasm is the jelly-like substance inside the nucleus, and finally, the chromatin material is present. Endoplasmic reticulum is connecting the nucleus to the nuclear membrane to the cell membrane. Okay, so this is all written over here. All right, and there are some endoplasmic reticulum which bears the ribosome also. We will be discussing this in detail while I will be teaching you endoplasmic reticulum. Now let's see what is present inside the nucleus. As I told you, the DNA, genetic material is present. But do you know how the genetic material is present? So basically, the DNA is 2.2 meter long. Okay, it's so long, but this so much longer DNA is present inside tiny nucleus. How it is present? It's present in a super duper coiled and condensed structure. So here is the DNA. DNA is made up of these nucleotides, okay, and protein sequence are there. Proteins are there. So these are the histone proteins, okay. So this proteins plus DNA plus histone gets coiled to form the chromatin, and chromatin gets coiled to form the chromatids, and chromatid gets coiled to form the chromosomes. Okay, so basically uh, in a normal cell, regular cell, the cell which is at the interphase stage, the DNA is present in the form of chromatin. Clear? But when it enters the prophase stage, okay, like when it starts to divide, when it enters the M phase, so that time it starts condensing and converts into chromatids and then into the chromatid structure, basically the chromosomes, final chromosomes. Okay, otherwise normally the DNA is present in the form of chromatin when the cell is not dividing, it's an interphase stage. So now you can see there are some cells which are having multinucleated DNA. Okay, and there are some cells which are having anucleated DNA. That means they don't have DNA. Okay, so they don't have nucleus, sorry. So multinucleated means so the cells having more than one nucleus. For example, the cardiac muscles. Okay, they have so many nucleus. Okay, now, here you can see binucleate bladder cells. So, bladder cells are having two nucleus. Binucleate paramecium cell, again two nucleus are present. In the liver cell also binucleate, two nucleus are present. In the muscle cells, especially the cardiac muscles, multiple nucleus are present. Okay. So, now there are some anucleate, means nucleus are not present. For example, the RBC, erythrocytes, they don't have any nucleus. Okay, so that's about the different types, numbers of nucleus. Now, nucleoplasm contains the nucleolus here, and nucleolus is synthesizing the ribosomal rRNA basically. Okay, so they are synthesizing the ribosomes. Now, there are different types of chromosomes. Okay, according to the, see, this is the structure of the chromosome. First of all, this it has two arms, P arm and Q arm. Okay, this is the chromosome having two chromatids. So this is one chromatid, this is one chromatid. Both together are called as sister chromatids. Okay, so chromosomes having two chromatids. So basically, in the interphase, we have 23 pairs of chromosomes. Okay, so in the G1 also, you can go and check out the mitosis and meiosis topic. Uh, Check out that video. Okay, so here in the like gap 1 phase, this is the chromatid, how it looks like. This is the chromosome. But in the S phase, the number of chromatids gets doubled up. The amount of DNA gets doubled up, right? But the chromosome number remains the same. So it looks like this. It looks like this. So this is the one chromosome having two chromatids. This is one chromosome. This one is one chromosome. First one is one chromosome having one chromatid. I hope that this is clear to you. One chromosome having one chromatid, and this is one chromosome having two chromatids. Both are one chromosome only, not half chromosome. Okay. Now, at the center, this particular region is called as the centromere, where these two chromatids are getting attached. This region is called as the centromere region. Clear? Now, in some of the chromosomes, you can see that the centromere is at the center. So this is called as a metacentric chromosome. Some chromosome centromeres are is not at the center, properly at the center. It is like towards one, like moving in the upward direction. So that is called as the submetacentric chromosome. When the centromere is again moving towards the one direction, the end of the chromosome, that is the acrocentric chromosome. And when the centromere is at the top, like at the end only, that is the telocentric chromosome. So accordingly, we have different types of chromosomes. Okay. Now here you can see there is two terms called as satellite and the secondary constriction. What does these terms mean basically? So let's see this. So this centromere region where these chromatids are joining, this region is called as primary constriction region. Clear? This is called as a primary constriction region. So now in some of the chromosomes, like we have 23 pairs. So we in 13, 14, 15, while during this, formation of these chromosomes. So basically here also some uh, the chromosome gets narrower, okay, and there is also a constriction site happens. This site is called as the secondary constriction site. Okay, It is getting narrower basically, and a bunch of the part of chromosome is looking like a separate chromosome. And this is basically non-staining region. Non-staining. Okay. So here you can see this is the secondary constriction and here is the satellite chromosome. So this part of the chromosome is called as the satellite chromosome. Now this has a very important function. So secondary constriction is basically separating the, you can say it's separating the main chromosome from this satellite chromosome. This part is called as the satellite chromosome. Clear? So, or the chromosomes which have this kind of structure, like as I told you, 13, 14, and 15 chromosome has this kind of structure. Those chromosomes are specifically called as sat chromosome or satellite chromosome. Not all the chromosome has this structure, okay? Some chromosomes has only this structure. Now, this this thing, the secondary constriction region, okay, they are also called as nucleolar organizer regions or NOR. Why? Because they are helping in the formation of nucleolus during the interphase. And as I told you, what is the function of nucleolus? Synthesizing the rRNA. But what is the nucleolus? What is the nucleolus? See, this satellite chromosome, this satellite secondary constriction is like segregating the normal chromosome from the satellite. This satellite chromosome is going to form the nucleolus region, and that is going to contain this satellite region. This sequence contains the ribosomal rRNA, ribosomal RNA genes. So these genes are going to form the, basically these are the this region is going to create the nucleolus region, and that's how this region is going to be involved in the process of ribosome formation. Okay, so that means when I was telling you the nucleolus, who is creating that nucleolus region? These chromosomes only create the nucleolus region. And which chromosome? The satellite chromosome, satellite part of the chromosome. Okay, so here you can see the satellite located at the distal end of the chromosome. Secondary constriction region is located between the main body and the satellite chromosome. Basically, it's differentiating the two. Okay, satellite appears as a small separate segment, whereas the secondary constriction is a non-staining region on the chromosome. It is narrow and it is non-staining. So when we are staining the chromosome, we cannot see the secondary constriction under the stain. Okay. Satellite contains the genes which cause like the rRNA formation genes, and secondary chromosome is involved in the nucleolus organization. So basically these are going to form the nucleolus. So secondary constriction sites is the NOR, nuclear organizer region site. I hope this is clear to all of you. Okay, so that's till the nucleus. We have finished the nucleus. Now, the next cell organelle is the ribosomes. Ribosomes are discovered by George Palade. He observed the ribosome first under the electron microscope in the year 1953. Okay, now this ribosome, this this is basically involved in the synthesizing of proteins. So that's why they are also called as protein factory of the cell. Structure of the eukaryotic ribosome is 80S and structure of the prokaryotic ribosome is 70S. So S, what is the meaning of S? S is the sedimentation coefficient. So basically when you take the ribosome, put it in the centrifugation machine and it's rotating, so of course the denser and the less, a lighter particle will segregate. So the denser particle will settle sediment at the bottom, lighter particle will be at the top, right? That happens in the centrifugation. So here also when George Palade did this process, so he saw that some portion of the ribosome is settled down at the bottom And those are the he named it as a, like, it's actually it's heavy, so he measured it. He said that in the prokaryotic it's 50s and in the eukaryotic it's the 60s. Okay, and the lighter particles in the prokaryotic it's 30s, in the eukaryotic it's 40s. All right. Now, these subunits are not additive in nature, rather, they overlap together to form a complete ribosome. That's why 50 and 30 turns out to be 70s, 60 and 40 turns out to be 80s. Okay, so that is the basically the structure of the ribosome. Now, ribosomes are basically there. Some ribosomes are present as free ribosomes. Okay, so they are scattered throughout the cytoplasm, and some ribosomes are attached to the endoplasmic reticulum. So that is called as rough endoplasmic reticulum. So there are basically, we can say, ribosomes are present in two different locations, as a free in the cytoplasm and as a attached form also. Okay, these are the non-membrane bound organelles. Now, ribosome, here you can see they are helping in the process of protein synthesis, translation of the mRNA sequences into the proteins. Clear? So basically mRNA is the messenger RNA. Now messenger RNA, basically the DNA is sending all the message, okay, through the messenger RNA, and then ribosome is translating those sequence into proteins, whatever the protein is required in our body. So basically every parts of the cell, every parts of our body is the protein only, right? Because protein is the building block. So cell, like the cell membrane is having protein, lysosome is having protein. So these are all of these proteins are synthesized in the ribosomes only. Okay. So proteins produced in the free ribosome will function in the cell cytosol, while protein synthesis in the ribosomes bound to endoplasmic reticulum are secreted out of the cell, packaged into the organelles or inserted into the cell membrane. So the proteins which the free ribosomes are synthesized. Free ribosomes means the ribosomes which are present in the cytoplasm. Those proteins are functioning in the process which is happening in the cytoplasm only. Suppose there is a chemical reactions which are happening in the cytoplasm. So those proteins are helping in that process. But the ribosomes which are attached to the endoplasmic reticulum, those proteins are transported to different parts of the cell. Okay, so that's about the ribosome. Now let's talk about the endoplasmic reticulum. So endoplasmic reticulums are of two types. So basically RER and the SER. So RERs are the where ribosomes are attached, and smooth endoplasmic reticulum where ribosomes are not attached. Okay. So endoplasmic reticulum is a network of membranous sacs and tubules, and basically they are connecting the nuclear membrane to the cell membrane, and they because of this tubular structure now endoplasmic reticulum also helps in the uh, it gives a proper shape or structure to the cell. Okay, so it also helps in that mechanical support to the cell basically because of this network like tubular structure. Okay, now RER, as I told you, ribosomes are present, so it will of course help in synthesizing of proteins. Okay, and which kind of proteins? The proteins which will be transported to different parts of the cells, those proteins. Okay, so these synthesized proteins. So what will be the function of SER then? Smooth endoplasmic reticulum helps to synthesize lipids, phospholipids, steroids, and also helps to detoxify the harmful substances, especially in the liver cells. Okay, it also stores and regulates the calcium ions, helps in signaling and the muscle contraction. So these are the functions of endoplasmic reticulum. I hope this is clear. Now, next, the proteins are synthesized. Now, it needs to be transported or packaged. So the endoplasmic reticulum, it is having these tubular structures. It's like road, network of roads. The protein get transported through this tubular road-like structure. Okay, and where it is reaching? It is reaching to the Golgi apparatus. Golgi apparatus is like a packaging shop. Okay, so first of all, proteins are prepared in the factory, which is called as ribosome. Then from the factory to the shop, it is transported through the roads, right? So roads are what? Roads are the endoplasmic reticulum. And now in the shop, like if you want to wrap some gifts, so the shopkeeper, you purchase some gifts and shopkeeper also wrap it for you, right? So this Golgi is the shop and the shopkeeper, it's going to wrap it up. Okay, packaging, do the packaging. So basically, this Golgi apparatus is having the cis phase and the trans phase. So from the cis phase, the protein is entering, okay, and from the trans phase, it is moving out. So what is the meaning of packaging now? When ribosome prepares the protein, all the proteins are a kind of primary proteins only. Okay, now, this primary proteins needs to be packaged. Now, what is the meaning of packaging? So, they gets converted into the secondary protein, okay, and the secondary then converted into the tertiary protein or the quaternary protein, okay, quaternary. This process is called as packaging. So, proteins are getting packaged into tertiary and the quaternary form. Clear? This is the packaging. Now, the next step is going to be the labeling. It's not mentioned over here. You can mention it. Labeling will also happen. What is the meaning of labeling? Like, when you are wrapping a gift, you put a sticker also. You know, if it's your friend's birthday, so happy birthday from like your name. So you put a sticker, right? And if you want to give it to someone else, like suppose you are not going, your brother is going or someone else is going to her birthday, so you will write down the address also, right? So here also labeling. So labeling means there are some like carbohydrates or lipids. These are getting attached to the protein molecule. Okay. This is like the labeling or tagging. It's like an address. Now it is moving from here in this direction. The vesicles are there. Vesicles are going to carry this protein to that particular address. Okay. So that's about the function of the Golgi apparatus. Let's watch this video for a better understanding.
After ribosomes leave the nucleus, they will have the important job of synthesizing or making proteins. Outside the nucleus, the ribosomes and the rest of the organelles float around in cytoplasm, which is the jelly-like substance. Ribosomes may wander freely within the cytoplasm or attach to the endoplasmic reticulum, sometimes abbreviated as ER. There are two types of ER. Rough ER has ribosomes attached to it, and smooth ER doesn't have ribosomes attached to it. The endoplasmic reticulum is a membrane-enclosed passageway for transporting materials, such as the proteins synthesized by ribosomes. Proteins and other materials emerge from the endoplasmic reticulum in small vesicles, where the Golgi apparatus, sometimes called the Golgi body, receives them. As proteins move through the Golgi body, they're customized into forms that the cell can use. The Golgi body does this by folding the proteins into usable shapes, or adding other materials onto them, such as lipids or carbohydrates.
All right, so that's all for today's video, students. Okay, in this video today we have discussed about the cytoplasm, cell wall, nucleus, ribosomes, endoplasmic reticulum, and Golgi apparatus. We have discussed about these cell organelles. Other cell organelles are going to be covered in our next video. So stay tuned. I post videos on every Tuesday, Thursday, and Saturday. Okay. So if you like this video, please hit the subscribe button. Okay, and share with your friends also. And take care. Have a nice day, all of you. Bye bye.